Hello everyone and welcome back to Microbiology. This video is for my lab practicum and it's going to be on the 5 I's of microbiology. The 5 I's include inoculation, which is where you create a culture, incubation, which is when you create an environment that is perfect for growing your organism in, isolation, which is where you either isolate or separate your one organism from another, inspection, which is when you're going to be actually viewing your organism either macroscopically or microscopically, which you will find out later on in the video, and then the identification, which is when you actually find out what your organism is. You're going to be setting up biochemical tests to identify what this organism is. So in this video I'm going to be showing you all of those 5 I's, and then I will be conducting experiments that will follow them so that we can find out what our unknown bacteria is. So before you can actually conduct your experiments, you're going to want to do a couple things prior. And for example, you're going to want to do a couple things before you actually come to the lab. For example, make sure you don't wear open-toed shoes. It's more preferred if you wear sneakers just to help protect your feet, and wear clothes that actually cover your body. Don't wear like leggings or shorts or stuff like that. Preferably wear like jeans so you don't have any bare skin. We don't want you to get hurt in the lab by things falling or things splattering on you that could injure you, so just keep your skin covered. And you're also going to be wearing a lab coat, which will help, but just with that. Also, make sure that you don't have any electronics out. Make sure that they're put away in your bag, so that means no computers, phones, tablets, anything like that. Also, make sure that you remove all of your jewelry. This includes earrings, nose rings, necklaces, bracelets, stuff like that. Also, make sure that your hair is up either in a bun or a ponytail. You don't want it down falling on your face, because then you won't see when you're doing your experiments. So just make sure you do those things before you come to the lab. Now, when you first come into the lab, make sure you take your bag or your backpack, whatever bag you have, and put it in an area that's going to be safe and secured and away from your lab table. Then, wash your hands with soap and water thoroughly. You want to get any microorganisms or filth that you have on your hands off. Then, what you're going to do is grab a pair of clean gloves, because next you're going to be disinfecting your workspace. To do this, use germicide to clean your workspace. The germicide is a chemical that will kill or inactivate any microorganisms that are or may be present on the table. We just want to make sure that it's clean for our experiments. And then what we're going to do is use paper towels to wipe down the workspace. And when you're done with the paper towels, the ones that you used, make sure that you dispose of them in the biohazardous waste container. And then take your gloves off and put those in the biohazardous waste container as well. We don't know, they could have been infected with when cleaning. And then wash your hands again just to get the germicide chemical and any potential microbes that you may have picked up off. And then you're ready to put on your PPE for your lab. The first thing that you're going to be putting on will be your gown. And if goggles are needed for the experiment, you would then put those on, and then lastly you would put your gloves on. And then after that you can gather the materials that you will need for your experiment. For this experiment you're going to be doing many different things. You're going to be staining, you're going to be using sterile broths, and using the streak plate method, and doing biochemical testing. But before you can do any of that, you have to prepare your biological sample first. And in this video, I'm going to be showing you how to prepare a smear. But first, let's collect the materials. Alright, so these are all the materials you're going to need. This is a China marker. You're going to use this to label your slide with. You're going to need a frosted glass slide. This is where your smear is going to go. I don't know if you can see that, but it's right there. You're also going to need a plastic pipette dropper to transfer your water onto the slide with. You're also going to need an inoculating loop, which is this guy right here. This is what you're going to use to actually transfer your unknown bacteria onto the slide. You also need a bacto incinerator, which is this guy right here. that is going to sterilize your inoculating loop. You're also going to need a Bunsen burner, which is right here. This guy right here. Okay, I don't actually have a real back-to-incinerator and a Bunsen burner, so I'm just kind of using whatever is around my house to show the experiment. But the Bunsen burner is going to be used to heat fix your smear. And you're also going to need a slide box, which is right here. You're also going to need a biohazardous waste container, which is the tissue box in the back. Well, the tissue box is going to represent the biohazardous waste container. And then you have your slant culture right here, which has your unknown bacteria in it. So, alright, let's get started. So the first step in this experiment would be to label your slide, and you would do that with your China marker. I already did that step. As you can see, I already put my initials on there, and generally you would actually put the microorganism's name on the slide as well, but we actually don't know what our bacterium is, so I just put UB, which stands for unknown bacteria. And also, with your China marker, you're going to be creating a circle in the middle of your slide, and what the circle represents is the spot where your bacteria is going to be smeared on. I don't know if you can see that. Oh, there we go. But yeah, you're going to do that. I'm just going to put this back down. And once you've marked up your slide, you're now ready to actually begin the smearing procedure. So the first thing that you're going to do is you're going to use your plastic pipette dropper to transfer a small drop of water from this guy right here onto the slide down here. Now, in a real experiment, we'd be using a sterilized plastic pipette, or I wouldn't have it just laying around on the table. But because this isn't actually a real experiment, I can, well, do it. But just remember, it needs to be sterile. So, I'm going to take the water here, get a couple of drops from that, put that back down. And just going to move this so you can see it a little better. I'm just going to put one small drop in the center. You don't want too much water because this slide is going to eventually have to air dry. Now that you put your drop of water on the slide, it is now time to obtain our culture. But before we can do that, we have to sterilize our inoculating loop. So in order to do that, we would put it in the back-to-incinerator for at least 10 seconds. So I'm just going to be showing you a close-up of how I'd do that. So to sterilize your inoculating loop, you're going to stick it in like this. You don't want to stick the whole thing in because you don't want to melt the handle. You're going to stick it in like this and hold it in for about 10 seconds. Then allow your inoculating loop to cool off. If we don't allow it to cool off, it could potentially kill off bacterial cells that are sensitive to heat. So that's why we want to do that. Once your loop has been sterilized, you may now obtain a small amount of culture from this test tube right here. And to do this aseptically, you're going to take the cap off like so. And then you're going to scrape the culture out. You only want a small amount. Put the cap back on. Put it back on the test tube rack. And then what you're going to do is you're going to put it onto the drop of water that's on the slide. What you're going to do is gently rub it back and forth so that your unknown bacteria is spread evenly throughout the drop of water. Like so. Then sterilize your loop in the back-to-incinerator for about 10 seconds. And then set it back down on your bench top. Then what you're going to do is you're just going to allow your slide to air dry. That's it. You just want to let it air dry so that the bacterial cells have enough time to properly be arranged. So after your slide has completely air dried, you are now ready to move on to the next step, which is heat fixing it. And to do this, you're going to need a Bunsen burner, which is this right here. If you were in a real lab, you would turn it on, and then what you're going to do is you're going to pick up your slide, and what you're going to do is briefly pass it through the flame of the Bunsen burner 3 times. And then once you're done, just allow the slide to cool off. Just going to put it back down here. And the reason why we actually heat fix the smear is so that the bacterial cells stick better to the surface of the slide. Now, for this experiment, you're going to need more than one slide that has a smear on it. So what you're going to do is you're going to repeat the steps from where you took the drop of water, put it on the slide, obtain the culture from the test tube, and all that, and do it on another slide. So you have 2 heat fix slides, because later on in this experiment, you're going to need these 2 slides for an acid fasting stain and an endospore stain. So just remember that. And when you're done heat fixing your second slide, along with your first slide, what you're going to do is put your slide, I know this isn't a real slide box, but if it were, you would take your slide and put it in the slide box for safekeeping. Once your slide's been put away in a safe place, the slide box, you would then disinfect your workspace using germicide to, again, kill or inactivate any of the microbes that could be on this workbench. Of course, you would move all the materials out of the way so that you could disinfect it and clean it, and then you'd use paper towels to wipe it down and dry it off, and then of course you would, after you were done with those paper towels, you would get rid of them by putting them in the biohazardous waste container. And then after that, you would remove your gloves as well, these, and put them in the biohazardous waste container as well. And then once you've done that, you want to wash your hands thoroughly to get any possible microbes that could be on your hands. And then grab a new pair of gloves, because you're now moving on to the next experiment, which will be staining. Yay! More specifically, gram staining.